Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast with Richard Jacobs.
Before we begin, a note from our sponsor. I'm Richard Jacobs, Executive Director of the nonprofit Finding Genius Foundation and host of the Finding Genius Podcast. In late 2016, I was rear-ended at 65 miles an hour by a truck on the highway, which sent me off-road into a ditch. The impact of the collision gave me a concussion and other injuries. At the hospital, a CT scan showed that I had thyroid nodules, which turned out to be cancer. It was then, when I had a biopsy of my neck, that I realized even if I was a millionaire, I wouldn't want a second or a third biopsy due to the pain and the invasiveness of it. And appointments at that time for thyroid experts were 3 to 6 months out. And I was worried about dying now, even if that was irrational. So because of this, I've decided to raise money to conduct a literature review on steroids, on the causes of anxiety and depression, a condition that affects well over 50 million people in the United States and hundreds of millions worldwide. Our goal is to create a codex, a guide that reveals all possible treatments for anxiety and depression for people that live with the condition, or for loved ones that have it, as my wife and my son do. To find out more about our fundraiser, visit findinggeniusfoundation.org and click on current initiatives. And now, to our guest.
Hello, this is Richard Jacobs with the Finding Genius Podcast, now part of the Finding Genius Foundation. Our returning guest, Charley Lineweaver. He's an associate professor at Australian National University. And we're going to talk about cancer. Even though he's a physicist, he has some unique insights into cancer. So, Charley, thank you for coming back.
Thank you very much.
Yeah, if you would, so your background is as a physicist, as I understand, but how did you get involved with, looking into cancer?
Well, I started out as a cosmologist. I was analyzing the data from the COBE satellite on the cosmic microwave background, and my PhD supervisor at Berkeley was George Smoot, and he won the Nobel Prize for our work on the microwave background. And but in 1995, when people started to detect exoplanets, I followed that research very closely. And because I had lots of data analyst skills, I said, hey, they're not doing this right. They should be combining this data. And so I got into exoplanet data analysis. Now, interestingly, this is also just at the time when they found ALH 84001. That was the Martian meteorite that ostensibly had some fossil life in it. So life on Mars or ancient Mars had been detected. And so this prompted lots of money to go into astrobiology at NASA. And so astrobiology became a thing. So I almost uniquely qualified to study astrobiology because my dad was a high school biology teacher. So I had literally grown up with skeletons and models of DNA in the closet. So I knew much more biology than any cosmologist and much more cosmology than any biologist. So I called myself a cosmobiologist. Now, interestingly, at the same time, that's when I met Paul Davies, and he was getting interested in astrobiology as well. And so essentially you had two cosmologists becoming very interested in astrobiology. Now, the thing about astrobiology is that we study life not on 100 million year time scale or a million or even a billion. We study the whole history of life on Earth because we're trying to figure out when life got started, how probable life is. Those are the bread and butter of astrobiological research. And so that's what we got involved in.
Charley, quick question here. Yeah. Has there been any evidence of life found outside of Earth? And could it be confused with us taking it there? Bacteria riding along with our spacecraft and our people, and that's how we discover it, that it was from us.
That is that's called forward contamination, and that is a big worry because it's almost impossible to completely sterilize any spacecraft that goes to the moon or Mars. And so the worry is that in 10 years or 20 years, when we get very sophisticated, sensitive equipment on Mars looking for life, that we will detect the life that we ourselves have brought there. That's called forward contamination, and that is certainly a worry. And most of the effort is in reverse contamination, and that is people are worried about Martian viruses or bacteria attaching to a spacecraft and then being brought back. I am much, much less worried about that, basically because I was taught about the lunar program where they had quarantine for the astronauts when they came back from the moon. And the people in charge of the quarantine were essentially experts in wheat rust, which was the major problem that this institute had to deal with. And so they were essentially, so it was really a boondoggle, a waste of money. And it was just based on fear, I guess. So I suspect that when we get those instruments on Mars, we will detect the type of bacteria that we have brought there ourselves. The reason, what they should do to prevent that is to do a much better job at figuring out what are the types of bacteria that are on these spacecraft and in these institutes where they are building these spacecraft, and look at every nook and cranny, swab everywhere, and then say, okay, this is the type of bacteria that we cannot really remove. And so that's what we should expect on Mars. This is another good story is that about 10 years ago, some astrobiologists were looking for bacteria or life in the upper atmosphere. So they created a balloon with a little box in it. And the idea was the box would go up into the atmosphere, let's say 40 kilometers, open up, collect a sample, close down, and then bring it back to Earth. And they did this. And when they analyzed it, they indeed found bacteria. But what they found was the bacteria that was all around the launch site, because the bacteria had gotten onto the edge of the equipment. When they lifted the thing up, opened the door, that bacteria came into the box. When they closed it, they had essentially trapped the bacteria that was in the launch site, but they hadn't thoroughly sampled the bacteria at the launch site. And those are the types of errors that can easily be made. And it's easy to look in hindsight. Hindsight is 2020, but that's essentially what happens again and again. And so so you're right. We could find the life that we ourselves have put there. Another problem is that we don't really know what life is. So a lot of people think, oh, there's life and there's non-life, but that's not the question. That's not clear at all. The most egregious example of a boundary case are the most abundant organisms on Earth, and these are viruses. If you ask your friendly biologist, are viruses alive? You get half of them saying yes and half of them saying no. But I looked at this issue very carefully, more carefully, and the results are not half yes, half no. Rather, a quarter of biologists say yes, viruses are alive. A quarter say no, they're not alive. A quarter say I don't know, or it's uncertain. And the remaining quarter say that question is undefined because we don't know what life is.
Last time we talked about that, but this time I wanted to ask you, how did this progress to cancer, and looking at, cosmobiology to cancer?
So Paul and I were, deep in the weeds of, okay, how did life evolve? What were the first billion years of life, the next billion years? What happened during the previous 4 billion years? How did life change over that time? And that's what astrobiology is about, the entire history of life on Earth. And then at that time, the National Institutes of Health and the National Cancer Institute, they were getting lots and lots and lots of data. And so much data from genomic sequencing of cancer and normal cells that they said, hey, we are biologists. We don't know how to deal with data. Let's ask the physicists. They know all about numbers and data crunching. So one of the grants from the NIH, Paul and colleagues at Arizona State University applied for, and they got a grant. Now, but this was not, there were about 10 other centers around the U.S. who were essentially hiring physicists as numerical crunching guns to help biologists understand the data that was coming in reams. Now, interestingly, Paul invited me to be part of this. And instead, like most of these groups had, hey, we have a new physicist here who has a new instrument who can more successfully sequence these things, or we can arrange the data in a better way to understand it, or we have a better instrument of some kind. That's typically the role that physicists play in hospitals. But Paul and I, being astrobiologists, said, wait a minute, what is cancer? Paul always likes to ask fundamental questions, and so do I. And so we were saying, Instead of crunching numbers, we were asking fundamental, almost philosophical questions. What is cancer? And then it seemed we were reading the literature, and some of the older literature said, hey, cancer is an atavism. And they just kind of were waving their hands and they said that. And I said, well, that makes perfect sense.
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now back to the show.
What is an atavism, by the way?
Okay. The word atavism means essentially like great-great-grandfather or something in Latin. And an atavism is something, now, when you were, let's say, 3 or 4 months old inside your mother's womb, you had a tail. And in between your fingers, there was webbing. And when whales are embryos, they also have feet. So what these are aspects of the morphology of a multicellular creature, which used to be present in the adult form, but during embryogenesis, during the development of embryo, those features went away. For example, the webbing between your fingers that you had as an embryo got eaten away. It was got sculpted away. So now you have fingers that are separated. But earlier on, we had webbed feet, webbed hands.
Similarly, it's like kind of a biological anachronism or something.
It's sometimes called a genetic throwback or an anachronism. But importantly, what I'm describing is development and how in early development you have features that disappear. An atavism is when those features do not disappear as an adult. A good example is sometimes you'll have an adult horse with 3 toes. We know from the evolution of horses that they used to have 5 toes, then it went to 3, and then it went to 1. So when a horse is born and develops with 3 toes, that's an atavism. Another atavism is called supernumerary nipples. You know, right now you and I have 2 nipples, but some people have 3 nipples or 4 nipples, and it's just beneath the nipples that we have. That's called a supernumerary nipple, and we know that this is a feature of our ancestors because our ancestors had more than 2 nipples, kind of more like pigs or cows or dogs. So there's a whole series of things, maybe dozens and dozens. If you look up atavism in the Wikipedia, you will see a whole list of them. I won't go through the list, but anyway, atavisms are features from earlier times in phylogeny, earlier times in the evolution that have not been successfully suppressed and therefore appear in the adult form.
So you're thinking what? Cancer is a form of an atavism?
Cancer. Our model is called the atavistic theory of cancer, and that is when cancer cell. Now the atavisms I just described are morphological atavisms, and that is in shape, like tails and new nipples and things. But also the idea is you can have physiological atavisms. So, for example, you and I are breathing oxygen right now, but oxygen has only been in the atmosphere about 2.5 billion years. Before that, our ancestors couldn't deal with oxygen. And so if we have cells that revert to not using oxygen, but doing what we did before that, that would be an atavism. And in fact, it's useful to have this ability in our cells because when we exercise really, really, really a lot, we don't get enough oxygen to our muscles. They then change their metabolism and then they produce lactose. And so our cells themselves have this ability to deal with an oxygen-free atmosphere, and it's very, very useful. It's a holdover from 2.5 billion years ago because there was no oxygen then, and so all cells had to do this. But then when oxygen came along, a more efficient in an oxygenated atmosphere metabolism came along. That's what our normal cells normally do. But in certain circumstances, like too much exercise, then you revert to this earlier way. And so our model, our atavistic model says that these cancer cells have reverted back to doing things that our cells used to do a long time ago, but these abilities have been stored inside our cells because sometimes, occasionally, these old abilities are in fact useful even today. That's why they're still around.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
So what would be the point of, do you think that then cancer comes because of like a forced adaptation, and the way that they go is they go back to their previous arrangements from millions and millions of years ago? Is that what causes this atavism?
Well, let's think about the non-cancer atavisms. Let's say a horse, let's say a whale. Sometimes porpoises are born with legs. So what has gone wrong? Usually they don't have legs, but sometimes they're born with legs. So what has gone wrong? What has gone wrong is that there are genetic regulatory networks, essentially genes that they turn off the development of hind legs. Now, and then you have a normal porpoise. But occasionally something will go wrong with those genes, those suppressing genes, and then presto changeo, the leg develops like it did before the evolution of those repressors. So when the repressors are damaged, out comes the old stuff, essentially. Now, so cancer, we think, is damage to damage to genetics, it could be, or epigenetics, and that is the normal functioning of the cell is damaged in some way, and that causes the reappearance of the old way of doing things for the cell, just like the old way of walking for a whale was on having hind legs, and those come out.
Your thought is it's still a random process. It just affects these regulatory networks, and that's why the atavism appears? Or do you think it's more of a directed response to pressures, which causes adaptation, but it's a maladaptation?
Okay, so what you just suggested is right now, the consensus, the most accepted view on cancer evolution is the following. There's damage to the cell from all kinds of things, UV or chemical toxins, all kinds of ways to damage cells. And then that creates variety in the genomes of these cells. And then through a process of internal Darwinism, in other words, evolution among cells that are already in your body, that then they get selected for adaptive features, which are novel, and they then allow these cells to survive inside your body. Even they are your own cells, but they are acting bad, but they have adaptations that have evolved out of all this damage. That's the consensus model. It's called the somatic mutation model, and that's one that we're pushing back on. And the reason we're pushing back on it is because we think all of the adaptations that cancer has, the ability to spread, the ability to divide very quickly, the ability to create blood vessels that then bring the nutrients, all of these things that make cancer so dangerous are features, are adaptations that are pre-existing. For example, when you have an embryo that comes down the fallopian tube, or fallopian tubes gets fertilized, it then does something called implantation. And during implantation, this very young embryo has to send out shoots to then put in essentially arteries that then get implanted in the wall of the womb. And that's a very aggressive procedure that this little fellow has to do in order to get the nutrients from the mother. But that's what has to happen. So these genes that do this are very, very necessary, but they're only necessary during the implantation of the embryo in the wall of the womb. Yeah. However, but then they have to be suppressed. They say, okay, we don't need that tool anymore. Get out of here, and you suppress it. But if that suppression becomes damaged, then out come these abilities to do what the embryo had to do earlier. And that's what we think. In other words, all of the abilities of cancer that are so bad and so adaptive for the cancer are pre-existing rather than the result of evolution on the variation that has been produced by damage.
So, I mean, some cancers occur in children. I don't know if anyone's born with it, but, a lot of them occur later in life. Why would it occur when someone's 65 years old and not, throughout the rest of their life.
Well, cancers do occur at all ages, but there's a definite correlation for getting old. When you get more cancer is more prominent or more prevalent when you get older. There are some very specific cancers that children get. Typically, some of these cancers evolve. Your body right now is made up of cells. Some of them, like the lining of your intestines, have to do mitosis all the time because you're essentially digesting your inner wall of your stomach and your intestines, so it has to be replenished. So that means you have to have mitosis going on all the time. Or your hair is growing. So those are two examples. And when you take current cancer medicine, it is anti-mitotic. It means it stops the division of cells. And that process of needing the division of cells is what your hair needs, is what your eyebrows need, is what your gut needs, your gastrointestinal tract needs. It needs cell division in order to act normally. Other parts of your body don't need to reproduce as much. For example, some of your bone cells or some of your muscles, they don't need to have mitosis as much. Interestingly, cancer is more prevalent in the types of cells in your body where mitosis is allowed, where it is promoted. And so that's something that's understandable because cancer becomes a problem when it mitoses too much. And some cells in your body are already mitosing at a healthy level. But now, I should point out at this point, there's something called the Hayflick limit. Have you heard of the Hayflick limit?
Yeah, the number of cell divisions before the cell goes senescent and dies, let's say.
Well, it doesn't necessarily die. You start out as one cell and you end up as, let's say, maybe 1 trillion cells, maybe 50 trillion. Anyway, you have to go from 1 to 50 trillion, and then you have to stop dividing. Or rather, when you divide some more, you have to get rid of some. So the Hayflick limit tells you how many cell divisions, let's say on average, a human cell will go through. Now, the Hayflick limit is something that evolved about 1 billion years ago because you wanted to produce a multicellular organism, not some single-celled organism that had no limit whatsoever on its ability to divide. So the Hayflick limit had to evolve. That means mitotic suppressors had to be applied in specific regions of this incipient multicellular organism so that the thing was organized, and so you could have mitosis in your GI tract, but not inside your, cartilage in your nose or something. So all of this had to be regulated. All of it had to evolve. And when something goes wrong with that regulation, cells revert to what they were doing earlier. And that essentially means doing things also that our embryos do very early. So that there are two aspects. There's phylogenetically ancient, and there's ontogeny or development early. And these two seem to be correlated, as we discussed earlier, when you had webbing between your fingers as an embryo.
So are there specific atavisms that show up for certain kinds of cancers, or in general for illness? Or, how is it determined, do you think, how far back the retrograde evolution will go and what abilities will be unlocked versus others?
Well, people have tried this and tested this model. Essentially, cancer cannot go further back than, unicellular life, because cancer is a coordinated effort. When you have a tumor, those tumor cells are communicating with each other, and they're together building networks of blood that give the tumor nutrients. And so there is a single cell stage which goes through the blood, and then it goes outside of the arteries, and then it's metastasizes. When cancer metastasizes, it spreads. It spreads as kind of individual cells that then leave the circulation and then embed themselves in various places. So that individual life is also something that's still part of us today. For example, your nose cells don't migrate around the body, but your immune cells and your blood cells do. That migration as individual cells is part of the normal functioning of a human being. And that's something that you can think of as being a very old, very early regulated thing. For another example, when you and I make sperm cells, if you look at the cross section of the tail of a sperm cell, it's very much like the tails of protists. If you cut them, they also have these things called undulopodia, sometimes falsely called flagella. But if you look at the cross section, they are almost identical. So this is something that has been well conserved, and essentially that's what you're doing when you're making sperm, is tapping into a very old genetic pathway that is useful, and so you hold on to it.
Again, do you think that nature has a playbook, and there's only a certain number of forms that have been rendered throughout time, and that these atavisms will by necessity constitute some of these earlier forms? Or do you think it's open-ended and it can, atavism could be anything?
No, I wouldn't say, no, not at all. I wouldn't say that nature has a playbook. Nature has a history. Nature has a phylogenetic history during which lots of adaptive features, adaptive physiologies, adaptive forms have evolved.
Why have cellular memory then? I mean, how would these old abilities be called up? I mean, would that be a form of cellular memory, or do you think it's just random that they happen to go back in time?
No, they're called up when the newly evolved suppressors get damaged. Remember, in order to stop the development of a hind leg in a whale, you have to have newly developed suppressors that say, hey, you hind legs, you're going to be a whale. We don't need you anymore. Suppress. And so that's what happens in normal whales. When something goes wrong with that suppression, as there sometimes do, there could be a genetic mutation, and then the hind legs are not suppressed, out come the hind legs. Similarly, if you have a normal cell inside your body, it's doing its thing. But when you have damage, mutations of some kind to the suppressors, then out come the earlier abilities that the cell really knows how to do well, and then it acts like cancer does.
Yeah, but are there abilities that come out that were not part of that organism's embryogenesis? Are there any abilities that come that don't correlate, that could not have come from that organism, again, during its development?
That is a very, very important question. Our stance on this is that no, there are no new or novel abilities that cancers learn. And so far, the data seems to be supporting our model, and that is none of the abilities of cancer are novel. As a matter of fact, when you look, Hanahan and Weinberg wrote a paper about, here are the hallmarks of cancer. Now, the fact that there are hallmarks, there are common features of cancer, these are the things that are characteristic of cancer, all kinds of cancer, and in fact, they can all be associated with atavistic traits. Now, you are essentially you're essentially hypothesizing something called the somatic mutation theory, and that is, you're saying, aren't there any new ones? Well, if there were new ones, then all cancers would be different because they're evolving in different bodies and they should be independent. And they're having different.
They are different in that they're very heterogeneous. I don't know how far the heterogeneity goes, but
no. Here's how far the heterogeneity goes. First of all, the genetics is very heterogeneous because there's a lot of noise there because you're damaging all kinds of things. But the easiest things to damage during, let's say UV or chemical toxins are the most recently evolved features of the organism. Why is that? Because anything that's really, really, really necessary is highly conserved and protected from all kinds of onslaughts. The more newly evolved things haven't learned to be that protected, and so they are more susceptible to UV damage and chemical damage and other types of damage.
So again, so far you haven't seen anything that doesn't come up in the embryogenesis of that particular organism when it gets cancer, for instance.
Well, you shouldn't. You use the word embryogenesis, but that's only one of the places where, let's say, old physiologies are important. For example, there's something called the Warburg effect. Have you heard of that?
Yeah, the mitochondria damaged, and that's so at least the fermentation versus oxphos and makes cancer. That's how cancer respirates, supposedly. Yeah.
That's right. And importantly, here's the important point. The Warburg effect is when it does what you just said, but it does it in an environment where it has oxygen. Normal cells, when they are doing glycolysis, and then oxygen, they do it when there's no oxygen. The oxygen comes back, and the normal cells revert to aerobic respiration. But cancer cells, when there is oxygen, are acting like there is not oxygen. That's why it's called aerobic glycolysis. Essentially, that's a physiology that all cells had to do before there was a lot of oxygen around. So there again, aerobic glycolysis, or the Warburg effect, can be easily explained by the atavistic model as a pre-existing, earlier very useful, but currently only useful in non-oxygenic environments. And we sometimes have that, as I pointed out, with runners who just use up too much oxygen, and then their cells have to survive by doing glycolysis, but then they revert back to normal function physiology when the oxygen appears. Cancer cells do not, because they have lost this new ability.
One thing I need to point out is, a substantial part of what makes us is our microbiome. The microbiome would have its own, I guess, atavistic adaptations, and the microbiome trades metabolites with our.
Wait. Let me clear it up. When I'm talking about atavism, I'm talking in the context of a multicellular organism. Now, you and I are multicellular organisms, so are dogs, so are trees, so are fungi. These are packets of billions and trillions of cells that have organized, that have become differentiated. You don't have just one type of cell, you have many, many different types of cells. And that's why when something goes wrong with your muscle or your immune system, there are so many different kinds of cancers, because there are so many different types of cells in your body, because there's so many different things that can go wrong. But as you get more and more advanced stages of cancer, your cells seem to be reverting to fewer and fewer types of cells. That's why it's sometimes called de-differentiation. They're becoming more. That's why the stem cell hypothesis also fits into the atavistic model, because in some sense you are reverting to non-differentiated cells, the kind you had earlier on.
I'm just asking you to consider the fact that bacteria change and mutate tremendously, sort of viruses, sort of fungi, protists, they all comprise our microbiome. What about those? If someone has cancer, there's going to be an interaction, a trading of metabolites, who knows what else. Perhaps our microbiome can also experience, a reversion to earlier behaviors and earlier pathways that also influences cancer, not just our somatic cells.
Well, if you're saying that the microbiome...
I say this because our microbiomes are so important to us, and we're a holobiont, and we're constituted of them, and, they're pretty necessary, for health. That's why I say it.
Absolutely. You're absolutely right about that. But there's one thing you're forgetting, and that is the microbiome are organisms that can evolve. And they are individuals that do not have, they're not nearly as organized together as a multicellular organism is.
But they do make biofilms that are complex geometries.
Absolutely. You're absolutely right. However, there's a big, big difference between what you just described as the evolution of the microbiome to any extent of integration, whether they're very integrated, like in a film, or they're individuals, like in some other aspects of it, I'm sure. But the big difference is that when you get cancer, you die. You do not pass cancer on to anybody around you. It's not contagious. Microbiome is just like any other ecology, and that is it continues. And so if it comes up with a new adaptation, that new adaptation is passed on to the next generation. Cancer cells do not do that. They kill the host. Therefore, cancer cells are a bad model for a group of cells that are evolving. But that is
there are a few cancers that are contagious, like, especially from your neck of the woods.
Yes.
I know it's, the Tasmanian devils bite each other and they give face cancer. And then some of the sexually transmitted diseases also may carry cancer, HPV, et cetera. So there are some that do that.
That's right. There are very, very few that do that. And they're easily operable because they're essentially like separate organisms that have stuck on the face, and you can just cut them out and they haven't attacked the rest of the body. I don't think they metastasize. They are contagious, but they don't metastasize. And it's not something it's not central to our efforts on the war on cancer, because it's not something that humans get, or maybe they do get it, but very, very, very rare. So we don't need to worry about it. We need to worry about the things that kill human beings. And those are the types of cancer that I've been talking about.
Yeah. Question here. Do you consider cancer a separate life form, especially when there's, let's say, a primary tumor and metastases? Because again, it's changed to the point where it's very different from somatic cells, and it has its own, drive to survive and proliferate and all that. So perhaps it is its own life form.
Well, no, I don't think so at all, because life forms continue. And what we're talking about cancer is cancer does not, the kind of cancer that humans have to worry about does not get passed on to the children or spread around the neighborhood. It dies with the patient. The reason why that's important is because anything that has been supposedly selected for during the internal Darwinism of cancer does not get passed on. So in the next generation, this cancer that starts in your children, for example, in 50 years, will have started fresh and new. It will not have any advantage of the adaptations that evolved in your cancer, for example, or your grandfather's cancer, or your great-grandfather's cancer. Cancer is very different because it dies. It doesn't continue. What does?
Yeah, but if I get infected by a parasite, it's a separate life form, or I get a bacterial infection or a viral, who knows? But I can get these infections, and some of them may lead to the parasite or the bacteria killing me, but yet they still are separate life forms. So why couldn't cancer be a separate life form?
Because that parasite in you could kill you, but in order to not be killed with you, it has to spread before you die into another patient. That's the whole thing about viruses, bacteria, or parasites. They have to continue and reproduce. Cancers do not do that.
Right, but just because cancer doesn't appear to have a mechanism by which it can transmit out of the organism in which it infects, that's just one parameter, perhaps, of what constitutes life. If it has all the other parameters, why couldn't it be a life form?
Because continuation and reproduction and continuing evolution is what life is about. And if you have a life form that lives for 10 seconds and then just dies, you don't have a life form if it doesn't reappear inheritance is part of what I think life should be. Or rather, let's not talk about whether it's a life form. Let's talk about how is it similar and how is it different from the microbiome, for example, or from a parasite. And the main difference is that cancer does not leave progeny in the next generation. As I said, if your grandchildren get cancer when they're 60 or 70, it will not have inherited that from you or anything that evolved in you during let's say, a decade that you had cancer, but rather it has to start afresh. And the fact that it too will resemble your cancer is because its physiology is pre-existing in the human genome in the same way that forming a back leg in a whale is pre-existing in a whale.
Well, I know this isn't as strong, but, if I have cancer and it epigenetically changes me, which undoubtedly it does, and then I have children, perhaps they may be they may have a predilection towards cancer, or maybe it has actually changed. I don't know. Maybe I had testicular cancer, let's say, and it changed the underlying composition of my sperm, and therefore, when I have children, maybe now they have, at the very least, a predilection, or maybe they have a genetic disposition towards that. Right. Maybe it passes in that way, but not the actual cell itself.
I would start to agree with you here, because to some extent, we know that epigenetic changes are passed along, maybe even to the second or third generation, and these changes. Now, so that's why one of the reasons why we want to look at epigenetics in so much detail, because to some extent, when cancer cells are acting haywire, it's because their epigenomes have probably been changed or rearranged into patterns that existed earlier on in development. So yes, it can be passed on in that direction, but I would say not it. You're passing on. It's kind of like there are such things as genetic-based cancers. In other words, when you have mutations in the genome that don't produce the normal organism, but rather it makes you susceptible to cancer. Matter of fact, there are genes that make you more susceptible, and you pass those genes on. Yes, you are passing on a susceptibility to cancer. That's certainly the case. If that's what you're talking about, I agree completely. But if you're trying to talk about something that has evolved in you, new, and then gets passed on, that's something that is much more questionable.
Yeah, throughout my life, like I said, I've experienced, massive epigenetic change. So in that way, again, it's not a direct passing on, but maybe it's an indirect passing on somehow.
Well, I suspect that the epigenetic change that has occurred in you or in me or in any multicellular organism as it develops and gets older has already been choreographed, has already been selected for. And as a matter of fact, aging is probably a good example of that. The aging looks to be genetic. And so what's happening is you're not wearing things out. Your cells have, your genome has arranged such that the epigenomes change. So you no longer repair what you used to repair and you get older. This is genetically programmed senescence. And this too is an issue that biologists are wrestling with because there's something called pleiotropic antagonism, which essentially means that when you have a mutation and it helps you early and hurts you all, and if it does both good and bad, well, then the good is shifted towards the beginning of life and the bad is shifted towards the end. And that's why you die, because you have an accumulation of these bad things after you have reproduced. That's that's also the consensus theory of why we have genetic senescence.
So if I look at the twins that went into space, I forget their name, but one of them was in the space station for a year.
Yeah.
You know, they showed that he experienced hundreds of epigenetic changes, which a lot of them regressed. I don't know about all, but once he came back to Earth. So to me, that shows, okay, his body underwent a change of environment. There was a forced adaptation, which the body accomplished by epigenetic change. Yes. And then when those stimuli were removed, it reverted.
Yes,
so, I mean, why wouldn't cancer also be, again, a maladaptation?
Well, what you just talked about is an adaptation. That adaptation has to be organized. You're saying, okay, let's suppose that when you get into weightlessness, you get methylation of this position in your genome. And then when the weightlessness goes away, you get the changes back to where it used to be. That is an adaptation. And the reason that it acts like that is because in the past, all kinds of things happened millions of years ago, billions of years ago, and there were variation and then susceptibility to, an environmental thing. When that happens in the environment, I'm going to have this epigenetic change. That is an adaptation. And then that adaptation is controlled by some gene and it gets passed on to your children. And so that's what, if not only this identical twin, but take you, if you send it out in outer space, you will probably have some similar epigenetic changes that would be adaptive to that environment, or not just any space. Space is a really bad example because that's not something that we experienced in the past so much. But let's say when it gets hot
maybe
outside,
it's a good example because no organism has experienced, being in, let's say, zero gravity,
right,
in Earth's history,
right,
so far as we know. So maybe it's a good one because
well,
there was no precedent. So the adaptation to that, it may tell you something very.
Well, then you get into what's called the problem with the spandrels of St. Marco, whether something is an adaptation or is it something that just came along on the ride for something you're adapting to something else. So this, for example, let's talk about reading. You and I both read. I can see you have glasses. I have glasses, and we can both read. But writing is only about 5,000 years old. So the ability to read, how in the world did that evolve? And if you pretend that reading is something that's totally unconnected to previous existence of human beings, then you are presented with this anomaly. How in the world did we evolve so quickly to be able to read? But if you look at what you need to read, and that is visual acuity in a broad general way, then yes, visual acuity did evolve in the past and helped us, look for fruit hidden behind leaves and trees when we were climbing around trees looking for fruit. So in other words, these abilities are correlated with each other. And so that's the way you have to explain reading or writing or any new ability that we can do now. It's not completely new, although we sometimes pretend it is.
Significant. Okay, I understand. I mean, so with your model of cancer, what light does it shed for you? What new thoughts or ways to treat it? Or I don't know if that's your angle.
No. Well, what we're trying to do is understand cancer. And so the question is, what does that have to do with therapy? How does that help? Now, here's probably the biggest, most important misunderstanding of the model. Like yesterday, for example, I read a paper and in Science magazine about, hey, we now know we have a much, much better map of the protein-protein interactions in human beings and in cancer and how they're different. right? So according to the atavistic model, the protein-protein interactions that are in cancer evolved a long, long time ago. They get repressed in normal cells, and so we don't know about them much. But normal cells have this whole normal range of protein-protein interaction. So in this new paper, they discovered, cancer cells don't have the same ones, they have different ones. And so I know this because I've talked to hundreds of oncologists. They say, okay, this is a difference in cancer. Let's target those new things that we've discovered that cancer has that normal cells supposedly don't have. So what that means is, now in the atavistic model, normal cells have those early ones. They only show up latently, only show up during certain periods of development, but they're still there in every cell. But normal cells, when you look at them superficially, they say, they're doing this. Now, when normal cells lose the ability to do normal protein-protein interactions, they revert to older ones. So when an oncologist says, okay, now I'm going to target these older ones, the older ways of behaving or physiology are the most protected. They are very, very important to the cell, just like mitosis is important. And then along comes the Hayflick limit and says, you have to be suppressed. Now, if you lose the Hayflick limit, or if you lose the ability to do these normal protein-protein interactions, and you have cancer, and then you target that, you're essentially targeting all cells at the same time, in the same way that when you target mitosis, you're targeting all the normal cells in your body that need to reproduce. So essentially, when you target what you've just discovered in cancer, you're targeting the strengths of cancer, not the weakness. The weakness of those cancer cells is their inability to do the normal things. And that's what needs to be targeted. And that's what the whole strategy is, just the reverse of what normal oncologists do to target cancer. They find something new about cancer and they say, let's kill it. But then they realize, hey, by the way, I'm creating all kinds of problems, all kinds of side effects in normal cells. I wonder why that is. I guess it's not just the cancer cells that are doing this. That's the type of shooting yourself in the foot that occurs with almost all cancer therapy.
It makes sense what you're saying. I like it. My only worry is that when someone has cancer, they essentially de-differentiating back into a gigantic stem cell or collection of them. And if you target what abilities cancer has lost, what if that affects like all the stem cells in the body, or the immune system, or other systems that need to have varying abilities or changing abilities over time?
Well, the normal stem cells do have the ability to do the normal things later on. So I don't think now. So here's a therapy that we proposed in 2014. Now, you may know about the history of the immune system. There's an adaptive one. Crudely speaking, there's an adaptive immune system which has evolved in the last 500 million years. An innate immune system that evolved earlier on. Now, the adaptive immune system is what allows you to have antibodies that go around looking. It's a learning system for the immune system. The innate one is a genetic one that's not easily changed. It's like built-in vaccine versus, hey, this is a new development of a vaccine. That's essentially the difference between an innate, more fixed immune system and the adaptive one that's more flexible. If cancer cells in our atavistic model, cancer cells have reverted to a more innate, and so they do not have the ability to do adaptive immunity, or rather, they don't have the ability to talk to the adaptive immune system anymore. And most oncologists define this as cancer suppressing adaptive immunity. In our model, it's not suppressing adaptive immunity. It's just inability to deal with it in the same way that cancer cells don't do normal respiration when there's oxygen. It's just a shutting down of the more recently evolved features. Now, if that's the case, then here's the simple model. You take a person and as normal cells have access to the adaptive immune system. So you say, okay, I'm going to give you a vaccine. There's something bad here. Let's say it's some virus, and here's a vaccine for it. And when I give the normal cells the vaccine, then they're protected from this virus. So then I infect that body with the virus explicitly. Normal cells that have access to adaptive immunity can fight it off because you've already tested this. But the cancer cells, we think, will not have access to the adaptive immune system, and so they will be preferentially targeted. Why? Because they have lost the ability. You see the difference in the strategy between targeting the weakness and targeting the strength.
Yeah, I see what you mean.
Well, that's interesting that you see, because as I said, I've talked to dozens and dozens of oncologists about this. They are so fixated on targeting what they have just discovered about how cancer works that they are that's doing the opposite of the therapy that our model suggests. And again and again and again, and when I've presented this model to therapists, I can see them stumbling over this. They're trying to reverse it in their head, and they just can't seem to do it. And so I'm very, the thing that I'm most disappointed in is what's it called when you get fixated on a target fixation when you're a bomber. You just go in and in. And I'm trying to reverse this so that you're targeting what cancer cells, the weakness of them, and that is what they have lost and can no longer do. And that requires a little bit more thought, such as the experiment that I just proposed.
Well, so has this gotten to the point where you've spoken to anyone about, maybe setting up a clinical trial? And if so, what would that look like?
I've spoken to lots of people about this, but as I said, I'm running into a brick wall in most cases. Occasionally, anyway, I've run into a brick wall. But here's another aspect that we haven't talked about, and that is there is this atavistic model makes predictions about the ages of the cancer cell, of the genes that are responsible for the physiology of cancer cells. And our prediction is that the genes will be preferentially older than the physiology of normal cells. And that it has been borne out by something called phylostratigraphy. There's a guy named David Good and some others in Serbia that have looked at the ages of genes and then looked at the genes that are active in cancer, and they found that this is the case. So that's one piece of evidence that supports the atavistic model. But as I said, I haven't gotten, matter of fact, there's just an article that's going to come out in Bioessays probably next week or the week after about an oncologist who said, I just ran into this atavistic model. Wow, it's been developed by 2 astrophysicists. Isn't that interesting? I think they have something here. We should start paying attention. That's essentially the degree to which current oncologists have integrated this model into their thoughts.
Yeah, I find a lot of people are very resistant to other points of view. So, unfortunately, that's pretty universal.
Well, I don't mind it. It's just that we need to get more evidence, and it needs to be tested and taken seriously. I just wish they would more open-minded. One thing that can explain this is that oncologists are not trained in evolution. They know almost nothing about evolution. As a matter of fact, I would say because they're interested in curing people. People show up, they're dying, and they have to figure out a cure. They do not have time to worry about the evolutionary mechanisms behind the cancer. When there are some more theoretically minded people who are not in the clinic, they're not doing translational medicine, they're trying to understand cancer, they have developed this somatic mutation theory in which they use the word evolution in a very, very different way than the atavistic model. They say the cells are evolving inside the body, and that can explain the features of cancer. Our model says that's completely wrong. These features have evolved a long, long time ago. They're pre-existing. There's nothing new about them. And this is the therapy that this atavistic model suggests. Somebody should try it. But to a large extent, they haven't.
So going forward, what would be your next big step if you're able to get people to listen? Is it a clinical trial to test?
Well, I don't. I'm not qualified to do clinical trials or even think about them. What I am qualified to do is read the papers in which standard model has been invoked to explain the experimental data that they have gotten from clinical trials, and explain it in the context of the atavistic model in a much better way. And once I can do that, that might shift some of the thinking about the theoretical interpretations of the models.
Well, very good. Charlie, what's the best way for people to learn more about your work? Where can they go?
Well, I guess you can Google Charley Lineweaver, Charley, not an IE, but an EY at the end, Charley Lineweaver. And you can see a whole bunch of stuff. You can see my website at Mount Stromlo Observatory. You can also see some of the cancer papers we've done. We have 2 cancer papers this year, one in Bioessays and another in a book about current research in cancer. Those are available on my website. Also, by the way, I think we haven't talked about extraterrestrials, but I've just created a course called arewealone.us, and it's online. So if you just search for arewealone.us, you should find the website that I've created. Essentially, I just went around to conferences, astrobiology conferences, asking the world's experts, are we alone, and why yes, why no, etc. That's where I did the research on the viral surveys of biologists and came up with those numbers I gave you earlier.
Well, very good. Well, Charlie, again, thanks for coming back. And I love to argue with you. It's fun, and I think we both learned something. So I appreciate you coming back to the podcast.
All right. Well, thank you for having me.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.